Darshana gave it away, but we will now move to the enzymatic machinery that forms the backbone of these functional responses that we just discussed. I welcome Cynthia Liu, research associate in the group, to present the immune cell mitochondrial enzyme changes and how they correlate with psychobiology. Welcome, Cynthia.
Okay, yeah. Thank you, Darshana. So today I'll be talking about immune cell mitochondrial phenotypes, so like related to what Jack and Anna have already presented, as well as how they relate to mood disorder symptoms and adversity. Okay, so beginning with a little bit of background. So as Jack and Anna have already mentioned, immune cells do have distinct mitochondrial and OXPHOS profiles, and this has been shown by a number of studies with various types of data. And the immune system is also relevant to mitochondrial disease. So as Anna has already mentioned, respiratory tract infections are a leading cause of death in mitochondrial diseases, which does suggest that there is some immune dysfunction with mitochondrial disease patients. In mitochondrial disease patients, we also have purifying selection of the disease-causing mutations, specifically in immune cell types. So we don't really know what the functional impact of these purifying selections are and how it might relate to the immune system dysfunction. And so one way of profiling and assessing the function of these immune mitochondria is by measuring the mitochondrial respiratory capacity, which is like measuring the respiratory capacity on a per mitochondrial basis, which can be done in tissues or cell homogenates, as we do in the immune cell subtypes here. And so in this study, we have measures of mitochondrial respiratory capacity, which include complex 1 activity, complex 2 activity, and complex 4 activity in the electron transport chain. And we also have measures of mitochondrial content, so that's citrate synthase, which is part of the TCA cycle, and also mitochondrial DNA copy number. We can then combine these measures into a mitochondrial respiratory capacity index, or MRC index, which scales the measures of OXPHOS complex activities over the measures of mitochondrial content. And so in MiSBIE, I quantified these individual variables, so complex 1, complex 2, complex 4, citrate synthase activity, mitochondrial DNA copy number, as well as the calculated combined MRC index. And we did these in specific immune cell subtypes, so monocytes, lymphocytes, neutrophils, PBMCs collected from 2 time points during the day, as well as platelets. And this was done in both healthy controls as well as the mitochondrial disease patients. So both those with the 3243 mutation as well as those with the single large-scale deletion. So first, we confirmed that immune cells do indeed have specific mitochondrial profiles. And we can see here that both the individual variables, so complex 1, complex 2, complex 4, and so on, as well as the combined MRC index in the bottom right corner, they're all distinct across specific immune cell subtypes in healthy controls. We can also see that the ratios of each individual variable is distinct across cell types, so demonstrating that each cell type has like a different kind of mitochondria. It's not as if all the enzymes are low for one cell type and then high for another. Some cell types have like higher complex 2 compared to complex 1, for example, and others have the opposite. So if we look at the middle panel over there, monocytes have a greater complex 1 compared to complex 2 ratio, whereas lymphocytes have the opposite. And we can also see this when we just like plot the ratios of the variables. So in the middle panel, for example, they're quite different, whereas other ratios and show different profiles. So then coming back to mitochondrial disease, we can see that the immune mitochondrial function is not actually significantly affected, which aligns with what Anna was talking about with the max activities before. So this heat map shows the average relative value for each variable and each cell type in mitochondrial disease compared to control, so both the mutation and deletion groups. And we can see that most of these are actually not significant, so the only significant difference is the complex 4 activity in monocytes, which is significantly higher in the mutation group. And just to note, this is not corrected for multiple comparisons. And then this lack of difference is also shown when we when we plot the combined MRC index between groups, we don't see any significant differences across cell types. And then when we look at the mitochondrial phenotypes, we also don't see significant differences between control and mitochondrial disease. And we can actually see that the differences between cell types are much greater than those between disease groups. So like the lymphocytes and monocytes between the control and mito D are actually like, they're closer between the cell types, and then they're separated like by cell type. And this is also reflected in the ratios. Not surprisingly so, given the lack of differences between control and mitochondrial disease, MRC, and immune cells, we also didn't see a correlation between the MRC and the individual variables of each cell type with the mitochondrial disease symptom measures, as well as the biomarkers GDF15 and FGF21 across the variables and across symptom measures, with the exception of maybe the platelets and with neutrophil complex 4 activity. And we can see this more clearly here with the significant correlations in the platelets and the neutrophils only, but across the other cell types, we do not see a correlation between MRC and the symptom measures. So what could this mean for mitochondrial disease? So yeah, immune cell mitochondrial function doesn't seem to be significantly impaired in mitochondrial disease, which means that other factors must be contributing to immune system dysfunction. So maybe this is to do with the stress response or stress hormones, which Natalia will be talking about later today, or it could be due to other stressors from the external environment, or perhaps psychosocial stress. And so now pivoting to psychosocial stress, so how do immune cell mitochondria relate to mood disorders and adversity? So a little bit of background on why we're interested in this. So the mechanisms through which mood disorders develop are not completely understood. However, we do know that both adversity and stress are risk factors, with both psychological stress and early life adversity being risk factors for the development of mood disorders such as depression and anxiety. Changes in the immune system are also linked to both adversity exposure and mood disorder development. For example, immune cell proportions are linked to adversity exposure and also the development of depression and anxiety. Adversity is also related to changes in inflammatory immune markers, and mood disorders are also related to changes in immune cell gene expression. With relation to mitochondria, they also appear to be sensitive to stress and adversity exposure. And changes in mitochondria are also seen with mood disorders. And it's also been shown that mood disorders and adversity interact to affect mitochondrial biology in whole blood and PBMCs. However, we don't know how specific immune cell subtypes are affected. And we know from what we've talked about earlier today, as well as other people's work, that immune cells have specific mitochondria. So given these relationships, we asked how mood symptoms relate to immune cell proportions and how immune cell type specific mitochondria functioning through measures of both MRC and also the seahorse data that Anna just presented are related. And then we also looked at how adversity affected this relationship. So first, looking at immune cell proportions, looking at the relationship of immune cell proportions with adversity exposure, overall we didn't see significant relationships between adversity exposure and immune cell proportion, with the exception of the monocyte to lymphocyte ratio and the platelet to lymphocyte ratio, which is positively correlated with life course adversity. Then looking at immune cell proportions and mood symptoms, overall we also didn't see any significant relationships between mood symptoms and immune cell proportions, with the exception, again, of the monocyte to lymphocyte ratio, which is positively correlated with both depressive and trait anxiety symptoms. When we looked at the impact of adversity on this relationship, however, we saw that exposure to childhood adversity significantly moderated this relationship between the proportion of neutrophils and lymphocytes with anxiety symptoms, as well as between the platelet to lymphocyte ratio with both depressive and anxiety symptoms. This can be seen more clearly when we looked at stratified analyses. So we split the population into two groups, two ways. So first we split by average life course adversity exposure, so those with above average lifetime adversity measured by the strain questionnaire, and those with below average adversity exposure measured by the strain questionnaire. And then we also split by childhood adversity exposure, so those who did and did not experience childhood trauma based off of the childhood trauma questionnaire. So in line with the moderation analyses, we see that when we stratify by childhood trauma exposure, we only see significant correlations between like the certain immune cell proportions, so neutrophils and monocytes, with the mood disorder symptoms. And these relationships we only see in those who did experience childhood trauma and not in those without. And so then moving on to immune cell specific mitochondrial respiratory capacity and how they relate to adversity and mood symptoms. With regard to adversity exposure, we saw negative correlations between monocyte MRC and life course adversity. And in relation to mood symptoms, we saw negative correlations between lymphocyte MRC and state anxiety symptoms. When we looked at the moderating effect of adversity on this relationship, we saw that childhood adversity actually moderated the negative relationship between lymphocyte MRC and mood symptoms, and that lifetime adversity also moderated specifically the relationship between depressive symptoms and lymphocyte MRC. When we stratified by adversity exposure, in the same way that we did with the immune cell proportions, we see much stronger negative correlations between lymphocytes and mood symptoms, lymphocyte MRC and mood symptoms in the high adversity exposure group only, and not in the low adversity exposure group. We then repeated these analyses with another measure of mitochondrial respiration, so the ATP-linked baseline respiration, or mito respiration here, which Anna presented earlier today. And this was just a note that I only have data for lymphocyte, monocytes, and neutrophils. So with adversity measures, we didn't see any significant correlations between mito respiration and adversity measures. But with regard to its relationship with mood symptoms, we again see a negative correlation between lymphocyte mito respiration and anxiety symptoms, and no other significant correlations were seen. With the moderation analyses, we also see a significant impact of lifetime adversity on the relationship between lymphocyte MRC, mito respiration, and depressive symptoms. So just a reminder, with the moderation analyses of between mito respiration and mood symptoms, we saw a significant impact of lifetime adversity on the relationship between lymphocyte mito respiration and depressive symptoms. And then when we did the stratified analyses, we saw a much stronger negative correlation in the high adversity group between lymphocyte mito respiration and depressive symptoms, which was not seen in the low adversity group. So this is just a summary of the results that I presented in the second half of this talk. Some key takeaways include that lymphocyte mitochondrial respiration, measured by either mito respiration or the MRC, are negatively correlated with mood symptoms, and this seems to have a stronger effect in those who experience greater adversity compared to those with lower adversity. And so what could this mean for human health? So we know that stress and disease are related, and our work in the lab has been looking at how mitochondria might be a way through which stress relates to disease and health outcomes. So here we just tested a small part of this, and we suggest that mitochondrial biology is relevant to both adversity and the development of mood disorder symptoms. So thank you.